Good day, everyone. I'm Teacher Habib, and I'm thrilled to meet you all and explore the fascinating world of biology together. So let's get started. So, on my contrary, here I'm going to talk about mitosis. Now, when we talk about mitosis is the growth in asexual, like in multicellular organisms, because cells are constantly wearing off and have to be replaced. Like so for us to explain mitosis, we're going to talk about the cell cycle, which includes the interphase. We have the G1 phase, the L phase, and the G2 phase, which I'm going to talk about. So let's get started. As I think I've explained everything you need to know about mitosis. Thank you all. So when we talk about mitosis, we talk about, just like I said, we talk about the cell cycle. Now, the first stage we have in the cell cycle is the G1 phase, called the gap phase. The gap one phase, G1 phase. But first, we all know that a cell, this is a cell. A cell contains of a cell membrane, the cytoplasm, which is all this space here is called the cytoplasm or the cytoplasm rather. I also have the nucleus. The nucleus is the powerhouse of the cell. This is where all the whole genetic material contains. So that's what we need to know about the cell. And for us to, when we talk about mitosis means like a cell. We have a cell. This is cell. We all know that we have the cytoplasm, the nucleus, and the cell membrane. That's what I said. And we also have organelles like the mitochondria, the endoplasmic reticulum, and some other stuff. And this same cell, we need to clone it, just like we need to replicate it, such that we're going to have two identical cells. And we know that in the nucleus we have 46 chromosomes, which 23 maternal and 23 paternal. So we have to divide it such that each will have 46 in each, which is like 2n, where n is 23. If n is 23, we have 46 in each. That's just what we need to do to have the cell replicated. Now that's what we know as mitosis. So for us to do that, we're going to start with the G1 phase, like I've said. So let me just clean this and let's continue. So the gap one phase. So here, for us to replicate a cell, we have organelles. This is what I said, like the mitochondria, the reticulum, the ribosomes. So let's just go with two, which is mitochondria and ribosome. So we need to make more organelles so that by the time we divide the cell, we are going to have enough organelle in each cell. So that means in the gap one phase, we have to make more organelles. That's the first thing we need to do. And the second thing here is to, like, we have to synthesize proteins and enzyme. You understand, sir? Because enzyme help is like catalyst, like it helps to speed up the action. So we have to synthesize proteins and enzymes. So that is all about the gap one phase. So we move to the next stage, which is the S phase, which is also known as the synthesis phase. Now, this synthesis phase, now we're going to talk about the cell replication. This is how we're going to replicate the cell, like the DNA, like the nucleus. Because for us to replicate a cell, that means everything has to be divided into two, which includes the nucleus also, which also includes the DNA, which is in the nucleus. Let me say this is a strand of DNA, and we are going to open it such that it's going to form something we know as a replication bubble. Now, in this synthesis phase, we are going to divide the DNA to form a replication bubble. Now there are three, I think there are two polymerases. We have the type 1 DNA polymerases that are helping and they're doing a great job for this DNA replication. DNA polymerases. We have the type 1 and the type 3. So we're going to move to the next phase, which is the gap 2 phase. Now this gap 2 phase, this is where we need to enlarge the cell, because if something is small, for us to divide it into two is going to be more difficult than if the thing is more bigger. So we have to enlarge the cell. So we are going to, by that we mean by enlarging the cytoplasm. So we are going to enlarge the cytoplasm. That means, let me say this is cell, and this is the nucleus, this is cytoplasm, and this is the organelles. We are going to make it more bigger, such that we are going to be able to replicate it. We are going to be able to divide it into two equal halves. So when we replicated the DNA to form a replication bubble, now we have, by doing that, we divided the DNA such that we multiplied the chromosome. Instead of having 2n number of chromosomes, we have 4n number of chromosomes, such that when n is 23, we're going to have 96 number of chromosomes. So by the time we separate it back, we're going to have, 92 actually. So by the time we separate it back, we're going to have 46 in each. So 46 plus 46, we have 92. So when we divide it, we also have 46 back in each. So in the gap 2 phase, this is where we align the cytoplasm so that we can be able to divide the cell. Now, the synthesis phase, gap 1 phase, the synthesis phase, which is known as the S phase, and the gap 2 phase, which is known as the G2 phase, makes up the interface. The three together makes up the interface. So I forgot to tell you guys that there are checkpoints. Here, like when cells are passing through the cell cycle, there are checkpoints that now I forgot to tell you. Now there's checkpoints because when cells are passing through the cell cycle from each phase, let me say from the gap 1 phase to the synthesis phase, you have to make sure that the cells are in order, like they are working well, like they are in good time. Like there are checkpoints, like there's a checkpoint here called the G1 to S checkpoint, because it's between the G1 phase and the S phase. It's called the G1 to S checkpoint. Now in the gap 1 phase, it's like cells there, they usually take long, about 8 hours to years in the gap phase, while in the S phase it takes about 6 hours, while the G2 phase takes about 2 hours for cells like to stay there. Now we move to the next part. During mitosis, the main part is what's known as PMAT. Where you have the prophase. For us to easily remember, I call it PMAT. Known as P stands for prophase, M stands for metaphase, A stands for anaphase, and this T stands for telophase. So now the cell are finally divided into. Why would you do it like this?
I don't do like this. I just say.
Just continue. Now, when the cell move to the first part known as the prophase, let me just get this wiped off so that I can write up here. When cell moves to the prophase, here we have a cell in the prophase. Now, within, just like I said, within the nucleus we have DNA, which are wrapped by histoprotein to form chromatids. Now, this DNA, they are loose, just like they are loose strands. Now, when they are in the prophase, they unloose to form the X-shaped chromosomes, which we all know as this. They loosen up to form the S phase, the S-shaped chromosome, which they will be here like this. Each but because there's no space, but like they will be into 46. They will, all of them will be piled up in the prophase. Now, if it's about the cell membrane, you know, at the cell they have what we call known as the nuclear envelope, which is uh, the part of the cell membrane. They all dissolve. That's why we can't see the cell membrane during the prophase part, and the and some organelles like the ribosome, the mitochondria, they form what's known as the MTOC, something like this, the MTOC, which is the microtubule organization center, also known as where the spindle fiber arise from. Now, during the prophase, just like I said, cell loses to form the X-shaped chromosomes. Now, we move to the other stage known as the metaphase. During the metaphase, this MTOC, they align here, while the chromosome align at the center. They will be aligned into 96, but because there is no space, let me just use one, or maybe let me just use one or two. They will just align like that into 96. So with the metaphase, chromosome align at the center. And now we move to the next phase, which is the anaphase. Now in the anaphase, the spindle fiber or the microtubule association center attach themselves to the center of this chromosome. They attach to the centromere of this chromosome. You see they'll just be aligned like that. Now, this is a chromosome, right? And the center is known as the centromere. Now, the centromere determines the number of chromosomes. So, this is also a chromosome because it has only one centromere. So, if there are chromosomes like this up to 3 and they have one centromere, it is still one chromosome because centromere determines what? The number of what? Chromosomes. So the center, there's a center in the centromere in which the spindle fiber align to, with which they attach with, which the center, there's a purple structure known as the kinetochore. The kinetochore is a purple structure in which the spindle fiber attached to in the centromere. Now, when they attach themselves to the anaphase, they begin to repel to like this spindle fiber or the microtubular aggregation center. They contain motor proteins. These motor proteins are sensitive, so whenever the spindle fiber attached to them, they will begin to withdraw backwards. They contain two enzymes known as kinin and dynesin. So they begin to repel, move forward, and that's where they separate. So when this, so like when they separate, which each of them will separate, they will now separate and move on to the next phase, which is known as the telophase. It's the part of the telophase that we have something known as the cleavage furrow. This place, like this telophase, this where the cell begins to split into two, just like this a cell, and begin to press it here, it now form the what? The cleavage what? Furrow. Now, but during this part, the chromosome loses back to form the egg, to form DNA in the nucleus. We have our nucleus back, they lose back to form the DNA. So this is the DNA, the nucleus, and the microtubular aggregation center comes back to their organelles such as ribosome on the mitochondria. We also have our ribosome and mitochondria, and these they are cleavage follow. Now the cell begin to what? Replicate. Have organelles here in the person will put down the organelles. So they begin to what? Separate back. Now they separate into two cells, two identical cells. We have our nucleus, our organelles, and that is how cell gets separated. Cell gets replicated to form two cells. Now, there are some cells that like passing through the cell cycle every time. They are known as example are labile cells like the epithelium of the skin, the GI tract, like this cell and hematopoietic stem cell in the kidney too, like in the liver, yeah. Like passing through the cell cycle every time. They pass through the cell cycle, they get replicated every time. There are some cells that don't like passing through the cell cycle every time. Sometimes they might pass through the cell cycle or they won't. So these cells are known as permanent cell. Example are the sorry for looking at example are the skeletal muscle cell, the cardiac muscle cell which is in the heart, and the neuron cell. They don't pass through the cell cycle. They just it like they will stay in a phase known as the quiescent phase, also known as the G0 phase. Yeah, this is where some cells, even some stable cells, those are the epithelium of the kidney tubules and the liver cell, which are the hepatocytes. They are stable cells. Some they stay in the quiescent phase if they don't pass through the cell cycle because it's only liver cells, which is the epithelium of the skin and the GI tract, the life passing through the cell cycle every time, every day, every day. Mitosis is happening every time, every day. Right now, it is about 24 hours. Yeah, we have 8 hours, like the first one, 6 hours, 2 hours. Here we have 1 hour. And also there's a checkpoint. Because again, here we have the G2/M checkpoint to check whether the cell is still stable. I'm sure there's a checkpoint here. But we just call this one the M checkpoint, not M stroke A checkpoint. It's just the metaphase checkpoint, the M checkpoint. So this is how cell is being replicated. So this is how mitosis work. We just need a cell and divide it into two, such that we're going to have 46. So here is 2m, 2m. Because when each divide, we have 2, 2, 2 each aligned into 46, aligned into 46. Together we have 92 of it. But I told you that the centromere determines the number of chromosomes. So when they divided, we have each 1, 2, 3, 4 into 46, and here 1, 2, 3, 4 into 46, and we also have 46 in each, where traditionally are maternal, traditionally are paternal. So this is all we need to know about mitosis. Thank you so much. Please hit the like button, subscribe, and follow for more. I also do teach on physics too. If you keep on following me, I will guide you through some other biology aspects. I also physics too. Thank you so much.